I would now like to introduce Alex back to the virtual stage to present our final topic for the day, analysis tools. Thanks, Alex.
Thank you. Let's do it. So already, Alba introduced several of the different tools that are useful for performing analysis of epitope data. And I'm gonna go further in that demonstration and discussion. And I just want to point out the crucial difference here between analysis tools that we're discussing today and the predictive tools that are going to be discussed tomorrow. So what we talk about here in the analysis tools is shown here. This is a shot from the IEDB website, and it's a variety of different tools that are designed to analyze epitope data that you might have because of your research or having mined the web or whatever. And you see that there are some here in the top part that are listed just as that, while some are listed with a flask. It's a lab denomination. What we mean by AR labs is that these tools are still being tweaked, but nevertheless, we wanted to make them available to the scientific community because we think they're quite useful, but they are not as extensively validated as the ones above. So let's take a look at some of these tools. Population coverage. So what the population coverage tool does is to extrapolate and calculate the fraction of individuals that are likely to bind or to respond to a set of epitopes. And in order to get this calculation, you basically have to provide two pieces of data. One is a list of one or more epitopes with known reactivity associated with one particular HLA type in terms of binding or restriction, and the particular HLA frequency in the responding population that you want to extrapolate responses in. So by analogy, what we have been discussing in several presentations, we do not maintain HLA frequencies, and we rely on external sources such as HLA allele frequencies.net. So let's look at an example. So here you have a fictional example where you have a bunch of different epitopes, and for each epitope, it is known that they react to some particular HLA type. And this, again, could be reactivity in terms of T cell or binding or whatever you want to extrapolate. And then let's imagine that you are interested in asking the question, what about this set of 11 epitopes? How would it react, for example, in North Africa, where there are all these different populations that you can select for your analysis? And so this would be the result, given the fact that you selected that particular set of epitopes with known reactivity and that particular geographical location and set of HLA frequencies. And this is the how is presented. So the coverage is defined as the number of individuals that would be expected to react to at least one of the peptide. And you see here it varies in the specific example from 70 to 60 as a low, 83 as a high, and on average 75.7% with this particular standard deviation. The other thing that is calculated is the average hits. So on average, how many peptides would the average person in this population recognize? And you see here, again, it varies from a low of 4.5 to a high of 8.7 with an average. The other thing that we show is the PC90, which is this red line. It basically is what fraction of the population would give a 90% population coverage. And you see what number of epitopes would be active in 90% of a population or more, and it's approximately 1 epitope. Here's another tool, which is the epitope conservancy analysis. And Alba already alluded how conservation and variation is a very important variable to consider in the context of reactivity and recognition by both antibody or T-cell adaptive responses. And the tool does accept as input a series of sequences which you are interested in determining the conservation of, and a set of epitopes that you want to know how well conserved or not are these epitopes in the target set of sequences. So the concept of conservation is also dependent on a threshold. So you could say of this particular set of epitopes, how many are 90% conserved, or at least 90% conserved in a particular set of sequences. And this is what is defined as the degree of conservation. So here, let's make an example. This, I believe, is LASA. Yes, it's LASA nucleoprotein. And so what you will do is enter your epitopes. This is a list of different epitopes that are of interest for one reason or another in this example. And then you are entering a set of sequences of a LASA nucleoprotein, which you are interested in interrogating to determine how well conserved are these. And then you select a identity threshold. It could be 100%, it could be less. And then you're going to do that. And so this is the result. So here you see these were the various different epitopes that we entered in the example with their sequence and their length. And then for each epitope is tabulated how conserved are these epitopes at the 100% level in the 64 sequences that we provided. So this particular epitope, for example, is found 100% conserved in 63 out of 64 sequences that we submitted, while, for example, this other epitope is a lot less conserved and is found identically conserved in only 18 out of the 64 sequences we provided for the analysis. So more details can be obtained by clicking. And if we click on this particular NP401 epitope, you will see the detail for each of the 64 different sequences that were provided, where any particular mutation is occurring, or obviously if it's identically found, then it's identical. But if there is a variation or mutation, it shows in red where and what is the difference that is detected. Another tool is the epitope cluster analysis tool. And this analyzes and groups sequences based on, again, a certain level of homology. And there are different ways to do this. And so we wanted to preserve the capacity for the user to select the approach that they preferred. And there are a number of different applications for this tool. And we use this tool a lot, for example, in some of the applications that Alba has described in making mega pools and eliminating redundancies, or also to understand cross-reactivity. Let me go into a little bit more detail in these three different approaches. So one is to cluster the sequences depending on a particular level of homology. So let's say, for example, the 70% homology. And so you have all sequences that have 70% homology as a link, but the drawback of doing that is that you may have that the peptide number 1 has 70% homology to peptide number 2, and peptide number 3 has 70% homology to peptide number 2, but peptide number 1 and number 3 do not have 70% homology. It's easy to visualize. For example, this is the case if you do overlapping peptides. If you have overlapping peptides spanning the entire sequence of an antigen, obviously the first peptide will have 0 homology with the last peptide. The approach is that you can generate interconnected clusters. And so here now you dictate that all the peptides in a cluster must have 70% homology with each other. And that is fine, but there is also a drawback. It is that one peptide can be actually part of multiple cliques, multiple clusters. So to walk the middle line, so to speak, we developed this other approach, the cluster break method, which we recommend, which is an extension of the first approach, but the cluster is broken down into subcluster basis on consensus sequence computation. So let me give you a particular example. So here it's some sequences as a matter of example: trapper, clapper, snapper, wrapper, time, daytime, anytime, per time, timeless, and so forth. So you can see that there is homology between the different sequences or words here, but it's how are we going to cluster them? So if you go and enter your sequences in the tool, you will then next ask to go to step 2, and you can select your threshold and select minimum lengths and things like that. And then you go to step 3, where you can select the method of choice. So when you do that, for example, in the all peptides, all connected peptides example, you will see the situation that I was referring to in terms of overlapping peptides. So here you see that trapper is actually clustered together with timeless, even though trapper and timeless really don't have any homology to each other, but trapper is homologous to wrapper time, and wrapper time is homologous to daytime, and daytime is homologous to timeless. And so that it could be a problem in depending on the particular application you're interested in. If you go instead to the fully interconnected example, the methodology will break it down. So now all members here have a homology to the consensus sequence, but as I was saying, you can have that the timer ends up into different clusters, is repeated, which is potentially a problem if you're trying to generate a cluster, for example, for a megapool and so forth. And so the cluster break method, we believe, solves some of these issues in the sense that it does generate these separate tools, but also allows you to visualize the relationship between the different tools, generate the consensus, and actually I'm not going to show you this because this is not a live demonstration, but you can actually click on the cluster and make it rotate and see how it's structured. Moving on to another tool is the rate tools. That is a tool that is designed to give some suggestions of what are the possible restrictions of a given set of peptides that have been tested in a HLA type set of subjects. And this is essentially conceptually an extension of a methodology that is used to calculate whether there is an HLA association, for example, with a particular disease or a particular trait. So what do? This is a lab designed tool still. We need to input two different types of data. One is the set of alleles that is present in the specific population in which the peptides have been tested, and the other is the response data. So how do it look like? So the allele data, as I was saying, this is a set which the tool works in sets of HLA typed subjects, and you don't need to have the HLA type for everything. You can have only class 2 or class 1 or both or whatever. But you see here, in this particular example, we have a set of donor 1, 2, 3, 4, 5, 6, 9, 13, and we have in each column the HLA type. So donor 1 is the beta 1 of 301 and 071 heterozygote, and so forth for DQ and DP and so forth. So this is the first set of data you enter. The second set is the reactivity. So here in this example, we have 5 different peptides with this particular sequence, but doesn't need to have a sequence. This is just for your own ease of seeing the data. Can be coded any way you want. And the reactivity is what is really crucial here. So for example, we see that this particular peptide here was reactive in donor 1 and donor 8 or whatever. This other peptide was positive in these 2 donors. This was positive in this donor. You can have missing data. So this particular peptide, for example, for one reason or another, was not tested in donor number 4 and 5 and so forth. So once you enter this second set of data, the odd ratios and response relative frequency of responses is calculated according to a formula. And so essentially what you are calculating is whether the responses are more frequent in donors that have one particular HLA type versus not. And here we can say, okay, there is a cutoff of 20 spots for a response to be considered positive, and we submit the data, and the data can be downloaded. And here there are also 2, this is the input and this is the results. There are 2 outputs. One is a filtered results that focus more on the positive outcome of the analysis, and then the complete report if you want to see every single value. And this can get very big, especially if you have a lot of donors, because there's a lot of different HLA types and a lot of peptides and so forth. So the complete report, it looks like this. So you have for again this particular peptide and this particular allele DPA101, A plus or plus says it means how many donors have this HLA type and responded. So in this particular case, there were 5 people that were DPA 103 positive and responded, and only 1 donor did not have this HLA but responded. But also, if you look at the ones that were not responding, 34 people had the allele, did not respond, and 10 people did not have allele, did not respond, for a total of 50. And so you can calculate the odds ratio and relative frequency, and this is not a significant association. And this is done for all different peptide HLA combinations. Now, if you are interested in just getting to the more interesting things, the things that are likely potential restriction, that is when you get the more restricted report that shows only the ones that have a response frequency greater than 1.3 and a p-value greater than 0.001. And we validated these thresholds based on experimental data that we had. But I want to point out that this is not Bonferroni corrected. But in this particular case, for example, this peptide, the gene T peptide, and the R-beta 3 O2 O2 allele, that's quite interesting because you see that of the people that had this allele and responded, there are 6 of them, while and 18 had the allele but did not respond, so the frequency is 6 out of 18. But if you look at the people that did not have this HLA, none of them responded. And in fact, people that did not have the allele and did not respond were 26. So this combination, if you do it exact Fisher, is actually have a very high significant p-value, and it's has an odds ratio and a relative frequency of about 2. So it's about twice as frequent the response in people that have an allele versus not. The last tool that I want to talk about is the immunogenicity predictions. So the deimmunization tool was designed to assist people that have an interest in not in predicting immunogenicity, but with the overall goal of getting rid of immunogenicity, eliminate immunogenicity. As an example, in the context of a drug that is meant to be used in people, and obviously you don't want the drug to be immunogenic because that could lead to problems and minimally to decrease of the drug effectivity and so forth. So that was just introduced as a background. And the approach here is once you enter the sequence, the tool will generate overlapping peptides spanning the entire protein sequences and then predict HLA class II binding regions. Apologize for the binding duplication here. And so in this particular case, you will have a one particular peptide that is predicted of being immunogenic, and then the tool will automatically look for substitutions in the sequence of a predicted immunogenic peptide that would be predicted to decrease binding. And because by introducing a mutation or a substitution in the predicted epitope, you can also influence the binding activity and immunogenicity of neighboring peptides. So you want to make sure that you don't accidentally eliminate one epitope and you create another one. So how does the tool work? Here you see in this particular case is put in EPO, and we chose a threshold for the immunogenicity prediction of 20, which is what we recommend. And then it will go through. Here you have 4 peptides that are predicted by the tool to be likely potential immunogenic peptides for HLA class 2. And then you can enter an email and to get the results. And at the end of the day, also the other point that I mentioned already is the fact that the tool calculates also whether in the 2 neighboring peptides, one before and one after, there is a likelihood of having created an additional epitope, and 1 is the best score and 10 is the worst score that you can get here. And here you see the output. This is the original peptide, and you see here all the different potential substitution throughout the sequences, and the difference in terms of median percentile of predicted immunogenicity or binding. And you see you want to select things that have a substantial decrease in median difference, which is corresponding to a predicted significant decrease in binding capacity, and at the same time do not create a epitope in the neighboring regions. So as a way of recapping here, I briefly mentioned some of the tools that are available on the IEDB to analyze epitope data: the population coverage, the conservancy, the cluster, the rate, and the deimmunization tools, all answering different aspects of the analysis of epitope data. Okay.
Thank you so much, Alex, for that presentation on the analysis tools. We're running a couple of minutes ahead of schedule, but we will move into the Q&A now, the final Q&A for the day. I can see we already have some questions coming through, so be sure to finish posting them, because we have about 15 to maybe 20 minutes for this last little section. So now I'd like to introduce Alba back with Alex. And Alba, would you like to take the first question?
I clicked on the first question, so I'll take it. There's a question from A. Rosenberg, and says, the data regarding vaccination to monkeypox all come from PBL. Is there difference in frequencies in lymphocyte in regional vaccination tissues? That is, in general, a very interesting question. There is absolutely no data in terms of human regional tissues that I am aware of. There has been data that has been generated, I assume, in looking at responses from non-human primates, where you have access to the regional tissue. In general, is very interesting to determine whether there is an immune response in the lymphatic or in the lungs or in general. Those are hard to come by, especially when you talk to humans. It's relatively easy to convince a mouse to donate spleen to life, to science, but that's not so easy for humans. There is some data that is generated looking at autopsies and so forth, but the short answer is that IEDB does not create data. We can only curate the data that is available in the literature, and in general, that data is highly interesting and also very scarce.
Okay, I'm gonna take the second question. Do you consider MHC restriction for Magapool design? And so the answer is yes, but let me delve a little bit more into detail. So when we design a Magapool, the purpose for us is to try to design something that can be broadly recognized in different geographical locations. So the approach that we utilize when we perform T-cell prediction is to perform prediction to a set of alleles that are actually very frequent worldwide. And so specifically, in the context of class 1, for instance, Bjorn has been showing you the T-cell prediction, and one point you might have appreciated is that you can select a reference allele, which comprises essentially the most frequent allele in the HLA frequency net. You can use that and then essentially guide your prediction toward alleles that are more frequent in the population. In the context of the CD4, we utilize a different approach, and that is based on promiscuity. So previous studies performed in the IEDB has shown that by selecting specific HLA class 2 alleles, if you run the prediction and these epitopes are recognized as a epitopes in multiple of these alleles, that suggests that they are very promiscuous. So essentially they can be presented to different HLA class 2 alleles in the population. And so we use these tools to actually make sure that we are taking into account region that we know could be generally recognized by the population. And Alex, I don't know if we want to alternate or if you want to go.
Yeah, let's alternate. So I'm going to go to the bottom so that so one is from Daniel Hakim. If an MHC class 2 restricted core epitope is highly conserved, but the flanking amino acid difference, should that still be considered a conserved epitope? Should I look for conservation of core epitope length 9 or conservation of full window length 15? Related, blast 70% is considered weakly conserved, blast 90% strongly conserved. How to close a match need to be to see similar behavior and justify experimental follow-up? All right, there's a lot to unpack here. And those are all very interesting questions, which are actually struggling or addressing experimentally as we speak. So first question, if the core is conserved, but the flanking regions are not, is this conserved? No, it isn't. In the sense that it has been shown by many studies that the flanking region beyond the 9 amino acids that engage the binding pocket can also be recognized by T-cell receptors and influence also binding. So I would not be entirely satisfied that even if a 9-mer core is conserved, but the flankings are not, I would not be satisfied to say this is a conserved epitope. The additional challenge is that is not, while there are methodology to predict which is the correct core of 9 amino acid that binds, that is also sometimes not entirely completely clear. So that adds an additional uncertainty. So I would certainly, we have been looking for class 2, we have been considering conservation on the full 15-mer. The next question is, what level of homology is cross-reactive? And again, there is no universal answer. There is a statistical answer. We have looked at many, many different instances where we actually know what is the sequence, and we actually looked at T-cell lines and clones and so forth. Today, what we would consider a 67% level of homology for class 2 and 80% level of homology for class 1 as reasonable threshold. But again, it's a statistical argument. There can be peptides that are less homologous that would be cross-reactive, and peptides that are highly homologous, but even a single substitution in a key MHC or TCR contact residus could completely obliterate recognition. Alba, back to you.
Okay, were you able to find epitopes unique to monkeypox and not vaccinia? And so I think this is a beautiful question, because while we haven't necessarily looked into that, I can provide you a way that you can actually answer the question yourself if you want by using IEDB. So if you want to find unique epitopes, you can essentially take the list of the monkeypox predated peptides and the vaccinia one and run a cluster tool. That will allow you to identify unique clusters that actually are not similar to each other. But then the question becomes how much those are conserved. And so you will have to take this list and then use the conservation tool and actually try to map that either to monkeypox or to vaccinia and see how conserved they are. The expectation in our hands is that you're not going to be able to find many unique epitopes due to the conservancy between monkeypox and vaccinia, but this would be a way you could address the question, and that could apply not only in the context of monkeypox. Alex, back to you.
So going back to the bottom, so Ritan Bhattacharya asked, there are times when the deimmunization submission output shows failed when there are several jobs running on different tabs. Can you elaborate why this happens? And second question from the same participant, is there any tool to quantify or predict that the protein sequence has become non-immunogenic once the deimmunization residues has been substituted? So great questions. One, if you have multiple jobs, already the deimmunization is fairly computational intensive. And that's why we offer the option of having the results emailed back to you. So while I have not encountered that specific situation, I would be inclined to imagine that if you have many, many different jobs going on at the same time, eventually you're going to break the bank. So perhaps a suggestion would be to do it sequentially and not try to do everything at the same time, or to download the IEDB and do it on your own machine. The second thing, is there any tool to quantify or predict that the protein sequence has become non-immunogenic? Well, to a certain extent, that becomes a circular argument that if you have introduced a substitution that is expected to decrease immunogenicity, then if you run an immunogenicity prediction on it, should be less predicted to be less immunogenic. However, I think there is a very interesting application in the sense that say that your protein has 3 epitopes predicted. What you could do is to run the deimmunization tool, find an optimal substitution to decrease the immunogenicity of epitope 2, and then put the sequence of this protein with the substitution decreasing the binding for epitope 2 in again, and find what is the substitution for epitope number 2 and so forth. So you could do that in somewhat iterative fashion. The other thing that you can do even better is say that you've done a certain substitution to decrease epitope number 1. You could put that again to see if you now have 2 substitutions, could you decrease immunogenicity even more for that particular epitope? Back to you, Alba.
Yeah, so I apologize for lack of clarity. Apparently, when I was talking about the megapool designing against monkeypox, there was some unclearance about step 2 and 3, and particularly the question is, what are the protein orthologs and how is the immunodominance determination carried out? So I would just like to try to backtrack and explain a little bit slowly the way we have been doing this. So we use the IEDB epitopes available for orthopox, and then we generate the ortholog sequence for monkeypox. So essentially we go from orthopox epitopes to monkeypox equivalent epitopes. At this point, because the nomenclature for the orthopox virus might not be necessarily the same across the different viruses, what we did was to map the monkeypox back to the proteome corresponding to the monkeypox sequence. And so this is what we meant with protein orthologs, because essentially not only you need to derive the sequence, but you also need to remap it to the monkeypox to actually get the equivalent protein. Then finally, how did we determine immunodominance? Once you do that, you're going to have a list of your epitopes and the corresponding proteins. So you can essentially count how many of these epitopes are contained in each one of these proteins. And so the way we determine immunodominance is essentially any protein that contains 5 or more epitopes, we consider it to be immunodominant. And so this is the way we have been doing the filtering to select the immunodominant protein that then we use for T-cell prediction. Back to you, Alex.
So, Adole Adina Mascot asked, can megapool design for monkeypox also work for chickenpox and other pox viruses? So there are two answers to that question. In general, orthopox viruses are highly conserved, and in fact, that's why, you know, originally it was found that the vaccinia from a cow could prevent smallpox, and we have also found that in general, the epitopes defined in vaccinia and other orthopox viruses are highly conserved in monkeypox and other pox viruses. Chickenpox is not a pox virus; it's a VZV, in which case then that was a completely different viruses, and I would not expect to be very high cross-reactivity. But also a quick question from Mauricio Calvo-Valer asked, any plans of a tool to increase immunogenicity? And probably not, in the sense that, or I should backtrack for a second. So increasing immunogenicity is probably problematic in the case of infectious diseases, because it is possible that you may have a substitution that increases binding. But if you now are trying to recall memory T-cell responses against another sequence, you might gain binding but losing cross-reactivity. Now, there is one set of instances in which increasing binding or immunogenicity is of interest and has been researched extensively, which is the cancer setting, where it has been shown that sometimes you make substitutions that increase binding or in any case alter the sequence, and that results in a greater immunogenicity. It's still unclear whether the effect that is called heteroclitic stimulation is due to just an increase in binding, or just the fact that you have a sequence that is a little different, and so you can break tolerance a little bit better. So that's back to you.
Yeah, so there has been a question about the monkeypox paper availability. So the paper has been accepted, but the paper is not out yet, and we hope it's going to be available soon. However, we have a previous version of the manuscript, and I would be happy to provide the link on the bioarchive. Instead, in terms of question, there was another one that is how you determine the optimal number of peptides in a megapool. And so this is an interesting question and has a lot of, essentially the real question is, what is your scientific question? There is not necessarily a straight answer to how many peptides you need in a megapool. The question is, are you interested in being comprehensive and take into account the entire proteome of a virus? Then probably you will need to change your cutoff, or you're gonna need to design multiple megapools because that's not gonna be available in a single one. Conversely, do you wanna be comprehensive and look very extensively to a single protein? Then that's the approach you're gonna use. So in terms of the optimal number of peptides, the answer is it really depends what is your scientific question. You wanna make sure to have a good balance in protein coverage and HLA coverage. You also don't want to have too many peptides inside a single megapool. In our hands, we try to not go above 300, 400 per each peptide pool, which is the point in which we start breaking a single megapool analysis in multiple pools. But in reality, that really depends on what is your scientific question. And so there is not just a straight answer. Back to you, Alex.
There is a question from Moshe Michael Appel. Is there a possibility for developing a deimmunization tool for B-cell epitopes, perhaps focused on specific isotypes, with a view of decreasing allergenicity for IgE epitopes? Very interesting. I think that that would be kind of the holy grail for the allergy field. There are some issues to be considered there. One thing that it's intrinsically predicting B-cell epitopes has been more difficult than predicting T-cell epitopes. And that is partially due to the fact that important and crucial antibody epitopes are often conformational. And so we've done work with some of the predictive tools that you will hear tomorrow have work essentially by projecting what is on the surface, and now with AlphaFold, obviously there is a lot of excitement. So that's potentially conceptually feasible. One issue with the antibody epitopes is the repertoire is very large, and so typically what has been seen is that if you remove a B-cell epitope or you change that B-cell epitope, if you make a substitution in a B-cell epitope, you may eliminate reactivity of one particular antibody recognizing an epitope, but another antibody can recognize the mutated epitope. And so as long as there is an active source of T-cell help, you will continue to get antibodies just to the mutated version. So that has been somewhat problematic. And in fact, that has been something that has certainly been debated a lot in the case of factor VIII, where utilizing factor VIIIs of different species so that antibodies would not cross-react led to development of a new inhibitory response. Back to you, Alba.
I think I have addressed most of the question at this point, Alex. So if you want to answer something, there are a few more related to your analysis.
Let me jump on. Do you use any, Jay Reddy asked us, do you use any criteria that should alert you to eliminate the papers from curating? Good question. I guess in eliminated papers from curating. So philosophically, the IEDB has taken the approach that it's not our job to give scorecards to papers. So it's not like, oh, this is a paper from my friend, I'm going to curate it for sure, or I don't like this paper, so I'm not going to curate it. And this is pretty much similar to what you do if you go on PubMed. You don't want your query to return the papers that the PubMed staff thought were pretty good. You just want to get everything that is published related to that particular topic. So we strive to curate all the papers that are past peer review, and our safeguard, so to speak, is that we curate the actual experiments on which that paper makes the statement that this is an epitope or whatever. So that enables the user to make that call. After saying that, there are instances in which we do not curate papers because they're not curatable according to our definitions. And a common instance is where the author does not really provide a complete information or understandable information. So, for example, I was mentioning earlier, if a paper says we tested a peptide from hepatitis B surface antigen 12 to 18, I don't know what sequence you made. There are many different sequences. And so we contact the author, but if eventually we can't determine what was done, what assay was used, or what was the sequence that was tested, we do not curate that data. I hope I answered your question.
Excellent. And I've given it a couple more minutes because the Q&A was flowing, but we are at 5 to 12. I think we'll answer a few more questions by typing as well. Is there anything else, Alex, you wanted to say?
No, thank you to all the audience for very good questions.